We get experimental science. We're curious, non-judgmental.
The Portia jumping spider.
Computer activity narrowed down the analysis to what seemed to be one brain cell. That one brain cell was able to put together the information from both sets of eyes.
He give it another brain cell. Biggest thing is where you would put it and how you would put it where you put it, because then that's not just knowing the neuroscience side of things. That would mean you would need to know the developmental biology, how exactly to direct these cell types of where to form and how to form. If you don't know that, we don't know, then adding an extra cell, it will just randomly appear in the brain. It wouldn't necessarily be one either. What you'd have to do is upregulate growth factors. You're essentially inducing almost a tumor-like effect into their brain because you're overactivating cell growth factors. Probably what would end up happening was it would make it so that it would just start forming connections elsewhere within the brain, and it might start forming misconnections as well, and then it might have a negative consequence. Add something to it with the spider's body fight against that cell. If you're using the genetics of the animal, the spider's body won't think that those are foreign cells because they'll be from the spider's body. It's from the same genetics. So there's nothing telling that spider that, these cells aren't necessarily supposed to be here versus the ones that are, because the outside markers of the cell still says this is me. So there's two kinds of signals. There is self and non-self. So self signal just says like, hey, I belong in here, I'm part of this organism. And then there's a second signal that says, I don't actually belong in here, I'm a bacteria, I'm a virus, or I'm something else. If you inject a drop of oil into a fruit fly larva, now a drop of oil, Sparky, right, is not alive, it's not a bacteria. There is no signal on the outside of that droplet that says, I'm foreign, and there's also not a receptor on that says I belong here. But the lack of those receptors actually cause there to be an immune response mounted against that oil droplet inside the fruit fly larva. So why would you try that, Aang? That's a great question. It's to simulate what happens when a parasitic wasp infects a fruit fly larva. Because the egg that they inject is about the size of an oil droplet, but when a parasitic wasp does this infection, they also inject venom. And the venom has its own effects that happen to the immune system of the animal. To remove that variable so that it's only be the object, like the size of the object, you would inject an oil droplet to start with. The ideal experiment, Aang, would be to just inject the egg by itself without the venom component. It's very difficult to dissect out the ovaries of a parasitic wasp and separate out the eggs and make sure they're fertilized and make sure that they're, gonna be injectable appropriately. Like, when they're in like a tube, so you're only gonna get a single injection. So there's like a lot of technical difficulties with it. The injection of an oil droplet is a good starting point.